get my sound on. Okay, so let me get you your notes for today. And we will put that away while we're doing notes. Okay, so let me turn the lights down. And like I said, we're not doing this whole packet today. We are just gonna get to a certain point and then we will switch gears and do our paper chromatography lab. Save some things for my people who aren't here. All right, so today and tomorrow we'll be focusing on the light reactions of photosynthesis, and then on Monday we will move on to the Calvin cycle. Light reactions are also called light dependent. This is my favorite picture for the light reactions just because I think it's the least confusing out of any diagrams I've seen. Sometimes it's called the Z scheme because it looks like a sideways letter Z. It is also called non-cyclic electron flow because I know we're used to a lot of things in biology, like the cell cycle being a cycle. This one is not a cycle. Okay, what you make at the end does not come back and go into the beginning. So we will end up calling this non-cyclic electron flow, but that'll be more important for you tomorrow. Okay, so we did all the background stuff yesterday. So today we've just got to hop right into the process. So photons of light energy strike a pigment in photosystem 2. And so I don't just want you to memorize one picture for a photosystem. You should be able to tell either of these pictures. I mean, that's just showing you part of a photosystem. But when you see those green circles, you know that those represent pigments. Same thing here. This entire picture is a photosystem. Each of the circles is going to be a pigment. And we learned about 3 of the main pigments yesterday. And what colors or wavelengths of light they absorb the best. And remember we said that we're going to represent a photon using one of those yellow zigzags. So when the photon of light strikes one of these pigments, it's going to absorb whatever wavelengths of light it can, but there might be other pigments around that can absorb different wavelengths of light. So that's why we're going to pass that energy on from pigment to pigment to pigment until it gets to the center of the photosystem, which we alluded to yesterday that's called the reaction center. So energy from the photon strike is passed from pigment to pigment until it reaches the reaction center, which includes a molecule of chlorophyll A in the center of the photosystem. Now, there could be chlorophyll A outside of that reaction center, because after all, it is a pigment that absorbs light energy. But in this reaction center, which is centrally located in the photosystem, that is definitely going to be a molecule of chlorophyll A in there. Okay, the thing about the reaction center that's important is that it houses a lot of electrons. Specifically, these are originally going to be electrons belonging to chlorophyll A. Eventually, we're going to have replacement electrons from a different source. So if we look at some of these pictures, remember I told you yesterday that there's two photosystems involved in the light reactions. Remember where all this is happening. This is all happening in the thylakoid membrane. That's where the photosystems are. So a photosystem is just a collection of pigments. This one that's a different color that's labeled, that's going to be your reaction center. So a photon of light is going to come in, it's going to strike one of the pigments. That pigment is going to absorb whatever wavelengths of light it absorbs best, and then it's going to pass that energy onto the other pigments because they might be absorbing other wavelengths of light. And then that energy from the photon strike is going to get to the reaction center. So the thing that you want to try and keep straight in your head over these next 2 days is that energy and electrons are two different things. The energy is from the light energy. Okay, what the energy is going to do is once it gets there, it's going to knock 2 electrons loose from chlorophyll A. So just keep electrons and energy separate in your head. This is just a different picture of a photosystem. Each of those things that looks like a lily pad is a photon, is a pigment. Photon of light comes in, it's going to bounce from pigment to pigment until it gets to that reaction center, which remember includes a molecule of chlorophyll A. So when the photon's energy finally reaches the reaction center, electrons there become excited and are raised to a higher energy level. Now, some of you in here have had chemistry, some of you haven't. Basically, it raises those electrons to an excited state. I don't really get into the whole ring thing in here. All you have to remember is that that's energy from light. It's a photon. So when it gets to the reaction center, it's enough energy that it's going to knock 2 electrons loose from that chlorophyll A molecule in the reaction center, and it's going to raise those electrons to an excited state. And so I kind of think of that excited state as like children on a sugar high, okay? We know that they can't stay up there for very long. They are not terribly stable at that excited state. And so electrons are the same way. When electrons are in that excited state, they don't tend to stay there for very long. They're going to get passed to different proteins in the electron transport chain, the first electron transport chain of the light reactions. Okay, I'll be right there. I'm coming. Hello. Sure thing, thank you. All right. That is you for early dismissal. I gave you both of your sheets, so you should be good. There's no homework in anatomy. All right, so photons knock two electrons loose from the electron cloud of chlorophyll A that is in the reaction center. Okay, so again, the photons will come in. Once they get to the reaction center, they're going to knock two electrons loose from chlorophyll A. So I told you that tomorrow we'll have these laminated posters that kind of function like a game board. And then I have these bingo chips that I've labeled as electrons and protons. And I think there's little foam pieces that are photons. And anyway, you're going to be moving them around. And so when you move the electrons around, you'll be taking, those electrons are going to be starting here, but when they get excited, you're going to raise them up to that excited state, and then the first molecule to accept them, which is technically still part of the reaction center of photosystem 2, is called pheophytin. Pheophytin is what's called the primary electron acceptor. Okay, so these electrons that were knocked loose, the first thing that's accepting them is called the primary electron acceptor, which is still part of photosystem 2, and it's called pheophytin. And then, so pheophytin is not part of the first electron transport chain. It is part of the reaction center. And then pheophytin is going to pass those energized electrons onto the first protein in the electron transport chain. So if we use that analogy of, kids on a sugar high, think about the analogy I've used for years is kind of like the photon would be like the ice cream man truck, or ice cream woman nowadays too, okay? And let's say he goes around, and he hits the neighborhood pool. And there's always gonna be kids at the neighborhood pool in the summer, and so all those kids get ice cream and they get super sugar high, and we know they can't stay at that excited state very long, and I guess the lifeguards or whoever's there taking care of them are going to pass those excited kids off from person to person to person to person. So maybe Grayson's in charge of them at first, maybe he's a camp counselor, and they go to a pool that day, and he's like, oh, these kids are too hyper, I'm passing them off to John. And then John's like, they got a little less energy now, but this isn't what I signed up for. And he passes them off to Noah. And Noah's like, they have a little less energy now. Still don't want to deal with this. He passes them off to Vera. Okay, so that's basically what happens during the electron transport chain is we know that those electrons cannot stay excited very long because they're not stable at that excited state. And so they will be passed to different proteins in the electron transport chain. And as they do, they are slowly losing energy. But remember what we said about energy in the last unit. We said that energy cannot be created or destroyed. It can only be transformed or transferred. So we are going to take that energy that those electrons are losing as they come down the chain, and we are going to use it for active transport to help hydrogen protons get actively transported into the thylakoid space. We'll get there. But first, Okay? It says if electrons from chlorophyll A kept getting excited by a photon over and over again, wouldn't photosystem 2 eventually run out of electrons? Because remember where those electrons originally are coming from. They're coming from a molecule of chlorophyll A that is in that reaction center. So you think photon comes in, electrons get excited, and they end up in that molecule over there. Next photon comes in, electrons get excited, they end up in another one of that molecule. And so you can imagine that if we kept doing this, eventually chlorophyll A is going to run out of electrons. So we've got to have a way to replenish these electrons that get excited. So while these electrons in that reaction center are originally from chlorophyll A, the replacement electrons are going to come from water. Now, when you see this, you don't have to draw that in because it'll be on another picture. It's on a picture a couple slides from now. But anyway, when we split water, we are splitting the hydrogen into its protons and electrons, and then the oxygen is going to come off. It's usually written as 1/2 O2 because oxygen's diatomic, which means you don't usually find just single oxygen atoms floating around in the atmosphere. So if you see 1/2 O2, it just means that that oxygen is going to be waiting for the next oxygen to be produced, and they'll combine to form molecular oxygen. But notice when it says 2E minus, that's for the electrons. They're going to go one place. The protons from hydrogen are going to go somewhere else. And so it's very important that you understand where those things are beheaded. So we're going to look at that on the next slide. Just a little bit, but I am okay. You can catch up on what you missed. And I do need your Scantron from your homework last night. All right, so it says an enzyme catalyzes the splitting of a water molecule into 2 electrons from hydrogen, 2 protons, H pluses, and an oxygen atom. So that the water gets split to form 2 hydrogen protons, 2 hydrogen electrons, and 1/2 O2. So remember what we mentioned yesterday. We said that when we talked about oxidation and reduction, we said we were going to track where the electrons went by specifically tracking hydrogens. And so that's definitely still true today. Okay, so then we're going to talk about where each of those things goes. Where do the protons go? Where do the electrons go? And what happens to the oxygen? So the electrons, they are going to replenish the electrons that were lost, that were previously knocked loose from chlorophyll A. Because remember, if chlorophyll A kept getting pairs of electrons knocked loose, eventually chlorophyll A would run out of electrons. And so to replenish that, we are going to split water, and the electrons from hydrogen from water are going to replenish those electrons in photosystem 2. Okay, H pluses will be released into the thylakoid space. And so you'll see a picture in a few minutes that'll definitely drive the point home that this whole process is occurring on or in the thylakoid membrane. So when we release those H pluses, since this is happening in the thylakoid membrane, it just means that the protons are going to be put into that thylakoid space, which is close by. So it's not like it has to travel far to get there. Next, we have oxygen. So remember that oxygen's diatomic. That's why we write it as 1/2 O2. It does not really like being by itself. And so oxygen is going to combine with another oxygen to form O2, which is the oxygen in the air we breathe. Okay, so think back to what we did yesterday. I drew the arrow in from water to oxygen, meaning a very common question to be asked is, from what molecule do we get the oxygen in the air we breathe? Okay, it comes from water. Okay, some people want to say it's CO2. CO2 is not even part of the Light reactions. It's part of the Calvin cycle that we'll do Monday. Okay, so you can't just look at something that has oxygen in it and say, oh, oxygen could have come from there. CO2 is not even part of this process. There's no CO2 here. So when water gets split, that's where we're getting that oxygen in the air we breathe. So remember what we did yesterday with oxygen and with oxidation and reduction. What's happening to water here as it becomes oxygen? Is it getting oxidized or reduced? Why? What does oxidation mean?
Lose electrons.
No? Oh, sorry, yeah, loss of electrons. I was getting my letters mixed up, sorry. Loss of electrons is oxidation, so this molecule has hydrogen, that one doesn't. Loss of electrons is oxidation, so we say water gets oxidized here. So I added this in below, you can add that in, or you can just write, does H2O get oxidized or reduced? It gets oxidized. Because in going from water to oxygen, water has hydrogen, oxygen doesn't. You are losing hydrogen, so therefore you are losing electrons, and that is oxidation occurring. And that would be something that once you kind of really cement it into your brain, that the oxygen in the air we breathe comes from the splitting of water. Okay, and then you look at the two formulas. You look at the formula for water and you look at the formula for molecular oxygen. That shouldn't be something you memorize. You should be able to look at that and say, well, water has hydrogen, that doesn't. It's losing hydrogen, losing electron, that's oxidation. Okay, if you try to memorize everything that gets oxidized and reduced, it may not be that hard in this unit, but it's going to be a lot harder to do that in the next unit. So I'd rather you just reason it out. Okay, so now that we have replenished those electrons, and again, people say, well, why is that step, that splitting of water step there? Shouldn't that be at the beginning? It could be. If you were to write an essay on this, maybe on your next test, I don't care where you put it. You either can put it at the very beginning, or you can put it where I put it in notes, which was talking about how you replenish those electrons. It just has to go in one of those two places. But now we got to pick up with what our electrons are doing. So next, the electrons will pass from the primary electron acceptor, which is thiophytin, to the first protein of the electron transport chain. Now remember where all this is happening. This is happening in the thylakoid membrane, and these proteins of the electron transport chain are embedded in the thylakoid membrane. So the electron transport chain is a series of membrane proteins. And this electron transport chain, because there are two, this is the first electron transport chain. It is made up of 3 proteins, and they are plastoquinone, cytochrome complex and plasticinin. Yes, you've got to know those. Yes, you've got to know that that's the order they go in, and that is descending order. Okay, so then we'll go to one of our diagram picks and we'll kind of go through what we've done so far, and we'll talk a little bit about chemiosmosis, and then that'll be it for notes for today. Just got a few more slides to go. All right, let's take a look. Again, this is the diagram I like because I think it's the easiest and least confusing. Photon of light comes in and strikes a pigment in photosystem 2. That light energy is going to get passed around until it gets to the reaction center, which is the little oval marked P680. The reaction center, remember, houses chlorophyll A and its electrons. And so when that photon of light gets there, it is going to be so powerful that it's going to knock 2 electrons loose from the electron cloud of chlorophyll A and raise them to a higher level where the primary electron acceptor, pheophytin, which is still technically part of the reaction center, will accept those electrons. Now, if we kept exciting electrons, we know that eventually chlorophyll A would run out of electrons. So to replenish those, we split water. And so the hydrogen, we split into a proton and an electron because it's only got one of each. Okay, but there's 2 hydrogen atoms. So that's how you get 2 hydrogen electrons, 2 hydrogen protons. The electrons are going to go right into that reaction center. The protons are going to go into the thylakoid space, which you'll see on the next slide. And the oxygen is going to come out the stomata so we can breathe it. And then these electrons, last we saw them, were at pheophytin, and now they're ready to go down the electron transport chain. So remember, when they're high up here, they're at that very unstable excited state, and they can't stay there for very long. And so when we do our poster activity tomorrow, yes, these have got to be shown in descending order because those electrons will be passed from pheophytin to plastoquinone. They're losing a little bit of energy. Then they're passed to cytochrome complex. They've lost a little bit more energy. So notice how this arrow over here says energy of electrons. When they pass to plasticinin, okay, so we've gone from pheophytin to plastoquinone to cytochrome complex to plasticinin, and then finally, when it's done with that first electron transport chain, those electrons are going to end up in the reaction center of photosystem 1. So remember, the photosystems are kind of backwards. Photosystem 2 comes first, photosystem 1 comes second. But the big deal is what happens during this first electron transport chain. Because I know back when I taught biology honors, kind of depends on who you had, but when I taught bio honors, I would just say, okay, that first electron transport chain, that's when you make ATP. I don't even remember if I talked about chemiosmosis in my honors bio class, okay? But we're going to talk about it here. And that is a big deal. And it's important to realize it only happens, this chemiosmosis step that we're going to talk about, only happens during this first electron transport chain. It does not happen over here at the second one. So let's look at a different diagram, which I still like. It's just kind of different. Now, I want you to get your bearings. First, fill in your blanks, and then we're going to look at what we're looking at here. We're going to identify some different parts. Okay, so what we said a minute ago was that as electrons are passed down the electron transport chain, they are losing energy. That's why you have me showing you in a downward kind of fashion, like going down a set of stairs. Okay, but we know that energy cannot be created or destroyed, so the energy they're losing has to go somewhere, and it is going to be used to make ATP through a process called chemiosmosis. And really that just involves building up an H plus gradient. Whenever you build up an H plus concentration, it is always built up in a space. So if you look at the picture, hopefully you can tell that this is the thylakoid membrane that you're looking at. When I told you light reactions happen in the thylakoid membrane, I meant it. Okay, there's your two photosystems. There's the first electron transport chain. There's the second. Okay, first the light reactions happen in the thylakoid membrane, and when you make ATP from this, you're going to make ATP out here in the stroma, and we'll talk about how you do that. But basically, just keep in mind, whether it's photosynthesis or respiration, when you build up an H plus gradient, you always build that up in a space. And so the only space we've talked about so far is the thylakoid space. So that means that's where we're going to be building up this H plus concentration, this H plus gradient. When we do respiration, you're going to build up that H plus concentration in the intermembrane space, which is between the outer membrane and inner membrane of the mitochondria. So let's look at this picture. Okay, it's showing water getting split. Notice where the electrons are going. They're going to replace those electrons lost by chlorophyll A. The oxygen can come off for us to breathe, but there's the two hydrogens from that hydrogen, the two hydrogen protons. Remember how I said that they are put into the thylakoid space, but that's just these two protons. I see another 4 protons there. They didn't come from this step. So we've got to go over where those protons came from. So photon of light comes in, hits one of the pigments, bounces around, gets to the reaction center, excites electrons, raises them to the higher energy state. It's accepted by pheophytin, which is the primary electron acceptor. And then those electrons are going to be passed to the 3 proteins making up the electron transport chain. And so first, they're passed to plastoquinone, then to the cytochrome complex, then to plastocyanin. And as they are passed from protein to protein in that electron transport chain, they are losing energy. But remember, that energy is not going to go to waste. We can do something with it. So we are going to harness that energy that they are giving up, and we are going to use it to actively transport H pluses from the stroma out here into the thylakoid space. So that's where these other 4 protons are coming from. They are coming from as these electrons lose energy, okay? If we're going to do active transport, we know that requires ATP. So we're going to take the energy that these electrons are losing as they come down that chain, and we're going to use their energy to actively transport H pluses from the stroma into the thylakoid space. Because remember, if you're doing active transport, you are putting something where there's already a lot of that something, and so it's going to require ATP to do so. And so it's in this thylakoid space that we build up an H plus gradient. When that H plus concentration is high enough, then the gate on ATP synthase, which is here, can open. H pluses will rush through. There really is a little turbine piece at the bottom. It's kind of like you saw in that animation yesterday. And basically that kind of works like a turbine. If those H pluses rush through fast enough, it's going to provide enough energy to add a phosphate group onto ADP to make ATP. The hydrogens, like I said, this channel doesn't open until the hydrogen concentration in there is high enough. And that's what will cause that ATP synthase channel to open. But notice, low concentration, high concentration, low concentration out here. So yes, it is going to take ATP to pump these 4 H pluses into the thylakoid space because there's already a lot of H pluses there. But once this concentration builds up here, there are way more H pluses in here than out here. So when that gate on ATP synthase opens, This is passive transport. This is basically just facilitated diffusion. They're going through that ATP synthase, which is a channel protein. And you do make ATP out in the stroma. So let's talk about chemiosmosis a little bit today, and then we'll pick up there tomorrow. So chemiosmosis also always involves setting up an H plus gradient. Hey, you know when you see H plus, they're going to ask you some application questions about pH, right? Okay, it's coming. If you don't know that when there's a lot of H pluses, that tends to be something that's acidic with a low pH, you got to brush up on that. Because they're not just going to ask you, you know, where is this occurring? And what I mean by that is, a lot of times College Board likes to ask you questions like, what if? So they'll say, well, what if you punctured this thylakoid membrane? What if there was a hole in it? Well, if there's a hole in it, then these H pluses that are in high concentration here, they're going to just naturally diffuse out. Well, if they just naturally diffuse out and I haven't built up an H plus concentration here, then that ATP synthase can't open and I can't make ATP. That may not be super clear to you yet, but I need you to be thinking about the what ifs. And then they could say, well, okay, if these H pluses would go through that hole in the membrane, then what's that going to do to the pH there? It's going to raise the pH in here because those H pluses are leaving, and it's going to lower the pH out here. So I need you to be familiar with pH, and on what end of the pH scale there's a lot of H pluses. All right, now the H pluses that we are trying to stockpile are going to come from two places. Okay, so these H pluses will come from two places. Water gets split on the side of the thylakoid membrane facing the thylakoid space, which will release H pluses into the thylakoid space. So I've shown you that in picture form. I just don't know that you've written it down in notes until now. Remember that H pluses are actively transported from the stroma into the thylakoid space. Okay, and so the next question says, well, if they're actively transported, I know active transport requires energy. Where are they getting that energy from? We're getting it from the energy that those electrons are losing as they come down the first electron transport chain. So you can flip to the next page once you have that filled in. I'm not going to go over that diagram again today, but I'll go over it with what we start with tomorrow. So we've got one more slide for today. I'm not going Okay, so this is everything that I told you in picture form. When the concentration of H pluses is large enough in the thylakoid space, that's going to trigger ATP synthase channel to open. And when protons go through ATP synthase, this is passive transport. This is just facilitated diffusion because they're going to be in higher concentration in the thylakoid space and then lower concentration in the stroma. So you've got to know when active transport occurs, you've got to know when passive transport occurs. And so once that ATP synthase channel opens, H pluses will rush through from the thylakoid space into the stroma because they're trying to get to the stroma because that's an area of lower H plus concentration. And so we say that that's an exergonic flow. And basically all that means is that those H pluses are rushing through that ATP synthase so fast, and there is actually a rotating piece at the bottom that will spin when those H pluses rush through, and that spinning creates enough energy to be able to add a phosphate group onto ADP to make ATP. Let me get you your lab. And like I said, lab we've got to do in here because I've got that bone marking practical set up in my lab. But I feel very sure that we can do this in here with very little difficulty. Wait till I go over the directions, and then you're welcome to go. Okay, we're not wearing goggles and aprons. I'm going to be handling any materials for you that we should be concerned about, so that won't be an issue. It says, today we'll be separating out the pigments found in spinach leaves, specifically spinach extract, using chromatography. And we will practice identifying which pigment is which, and we'll compare our RF values to known RF values. I don't know if we're doing that. But you're going to figure out how to calculate RF values. And so on the plate that I give you, there'll be a piece of TLC silica gel. When it says on this side of the lab, just understand we're not doing this in lab today. But the TLC plates, one side is like plastic and the other side is chalky. We are doing everything we're doing to the chalky side. It would not make any sense to do it to the plastic side. It'd be like trying to write with a pen, like a ballpoint pen on a piece of plastic. It doesn't usually work that well. So you're going to be doing everything to the chalky side. You're going to use a pencil, not a pen, to draw a faint pencil line 0.5 centimeter from the bottom edge of the TLC plate. And it says you are now ready to spot the TLC plate using a capillary tube. Basically, I'm going to give you a little cup that has the spinach extract mixed with acetone, and acetone is like the main ingredient in like nail polish remover. And I'm just eyeballing that. But instead of using a capillary tube or using a pipette, these came with my little kit. So I figured we'd try this. This is the little syringe like you use for catalase in our enzyme lab. And I feel like if you just suck some of the solution up in there, You should just be able to do a little drop at a time. You're trying to keep this drop hopefully smaller than a pencil eraser. And where you're doing that drop, that's what I wrote out to the side. Your spinach extract dot goes on the pencil line you just drew in the middle of that line. Okay, so on the picture down here, on the left, you see your pencil line, and then on the right, you see your little dot. Now, your dot may be a little bigger than that. That's okay, but it tells you to do that transfer about 4 times or so. So you're going to be doing like 4 drops from here. I need to save these syringes, so let's not throw them out today. Now, number 5, when you are ready for me, I will give you a beaker with chromatography solvent and a watch glass. I don't really want you handling that since we're not doing goggles and aprons in here. And essentially, it's just going to be like your picture down the bottom. The chromatography solvent is going to be in the bottom of your beaker. You're going to stick your TLC plate in there. It's going to lean against one of the walls of your beaker. But notice that the solvent has to be lower than your dot because the solvent is going to get pulled up that TLC paper, and then it's going to be separating out the different pigments in your spinach extract. And so I will give you the chromatography solvent when you're ready. And then you are basically watching that until your solvent front, which is, it's going to look like the TLC plate is like wet. When the solvent front gets within, where is that? Within about 1 centimeter from the top of your TLC plate, you're going to just remove the TLC plate from your beaker, and you can touch the TLC plate. That's not a big deal. And you're going to set it on a paper towel, and you immediately have to use a pencil to mark how far the solvent traveled, like just a horizontal line. And then on the next page, it's going to tell you how to do calculations. You're going to be using centimeters, so 4.1 centimeters, 4.5 centimeters, to measure first how far the solvent went from your original pencil line to your solvent front pencil line. And then for the other pigments, it's from your starting pencil line to wherever the pigment line is. And then it tells you how, below the chart, how to calculate the RF value. There is no units for RF because the units cancel out. And then you have just a reading to do with that paragraph, and then some questions to answer. So we're going to do this in here. We have, I don't know if we have even number anymore. 1, 2, 3, 4, 5, 6, 7, 8, 11, 12, 13, 14. All right. Yeah. Okay. So just, you just want to keep your lab out and something to write with, but put your binders off your desk and find somebody you want to work with, and I will come around and give you a plate. So if you need to use the restroom, I would go now before we get into this lab. Okay, so while you're waiting on me to get the spinach extract going, you guys can be drawing that pencil line. Do you have an odd number, or am I crazy? I don't know how many people went. I know Cadence is there. Are you coming to work over here, I guess?
Yes,
Oh. There you go. I only get 3 more plates. And remember, you are making your little faint pencil line on the chalky side. Okay, so there we go. And then And I've just got to make a spinach extract. I can't really make that ahead of time, because it will evaporate. So bear with me.
That's what she was here.
Draw a faint pencil line. Oh yeah.
Yeah.
It does say, it just says paint pencil line. Asking for I was just drawing that. It just says draw a faint line. It doesn't say draw. Make it straight.
All right, so this is your spinach extract. Remember, I know the directions say use a capillary tube or a pipette. I think the syringe will actually work a little better.
It's pretty faint if you ask me.
It ain't straight.
Cricket. Don't even just don't worry about it. Don't worry about it. It's fine.
Right.
So you won't need a whole lot up in your syringe because we're not really trying
But
to,
it's barely off. Okay.
It just looks
we're not trying to flood that TLC plate. And I'm gonna be pouring your chromatography solvent.
This groove. Like, that section. See, it's not irritating. It's
You don't say.
kind of irritating. I can't push it down.
So you're just transferring or doing those dots about 4 or 5 times, and that should be fine. And then I will get you your chromatography solvent.
Dip the capillary tube.
We're not using the capillary tube, you're using the syringe for that.
Yeah.
Your index finger is top.
It may spread to be a little bigger than a pencil eraser, that's okay.
So, that's some of the extra glue. Too. Transfer the TLC plate. So just pull it up, get some. You'd like to over.
Very softly. Once it gets going, it wants to keep going.
If you don't think the syringe works well, I can try some with my other glasses.
I don't know. You're trying to make a dot cup. Trying to make you're professional. We're supposed to be adding something to the paper. And then that was what we're supposed to be adding to the paper. Yeah, so I don't know. Hold on. You always
This all good?
Do we put a drop?
Yeah, we put a drop right here in the middle.
Look, yeah, but like it says, what's she talking about for?
Yeah. Yeah, there's
Put a
one.
dot, put a dot. Make sure you get it in the center, like in a random place.
No, you just put, I don't care if it's on the plate or not, put that in there and just put a watch glass over it.
Wait, you were supposed to put it in the same spot.
Oh my God. Yeah, now you're doing it over, because you didn't listen to what I said.
Why is it suction?
Look at the picture.
Wait, so,
Uh-oh,
oh.
that was a big drop.
Okay, so
That was a
there's the
little bit
LC
of-
plate.
I think that's a big drop.
You're gonna-
I did exactly what it says.
We're gonna see what happens.
That's a really, that's not the size of an eraser. You're a little off. No.
I'm not gonna touch it.
Yeah, I don't really, I think we'll just go with it. Here, y'all make sure you draw that pencil line in first.
I'm a little confused. You're very smart. Get the cover. Two. We're not using a cover two. You said we just have the storage transfer your TSC.
And I would put that watch glass just so you're not feeling all that.
Not to be enormous.
Push
We're not
your
your
little
dot to be
TLC
enormous.
plate in here,
I'm
just
not
have it
enormous.
lean against one side and then put the watch glass over it. You lean it, so like move the bottom of it, there.
Oh, we don't have the star 5 times.
So you're just gonna keep putting the dot right there.
No, you're done.
Yeah, I think we should ask
So
for
you just
dot. It's
lean
too big.
There's our dot,
There's
two
your
bars.
watch glass. All right,
Uh,
you good? You got it?
Is our
No,
No, you're fine.
Okay, good.
It will separate.
Yeah, there's. dot.
Basically,
Stick it
when we
in
get
the
to
tube.
thing, we just stick it in the tube and put the cap thing on it. We don't have everything.
So there you go.
Thank you.
And then you just lean, you're gonna put that in there kind of like that and then put that on the top.
I'll make sure you can mess that up. Okay.
I put it in this, right?
Now, where are you? Great. Let's keep reading. On both sides, bitch, you borrow Put your TNC plate, beakers. Okay, we did that. So, it's moving up.
I see.
It does, it was separated out the fans because it's not separating out our pigments.
No, it's different.
It's still green, although it's doing something.
Yeah, you see the bugs? Okay. You're done.
With the solvent from the solvent front leading edge of the solvent is about 1 centimeter from the top of your TLC plate. You should remove the TLC plate immediately to the pencil to mark how far the solvent travels. Like soon evaporate with the watch glass. Beaker to keep the beakers from evaporating. Okay,
Just let it sit
so we have to wait for this to get about a centimeter.
from the top.
So
How's it going over there,
double that.
Put this
From the top. We're gonna be there.
Oh, it's moving pretty fast. And it's like making a line. Look,
I know.
it like took that line off the thing.
What?
It's going. It made a line.
I'm not talking about the.
Distance moved. Measure the distance moved from your original pencil line. The pencil line you just drew. How far the solid traveled. It's gonna be the same. It tells us. It tells us to take it out. 1 centimeter.
So,
Oh, we do this a bunch of times.
what?
solve it. But we have all these that we have to do. It is making a bunch of line. We have dark green, we have light green.
And I know it says to put it, when you take it out and put it on a paper towel, we're just gonna put it on your plate. And remember that when you take it out, you have to immediately mark with a pencil where the wet mark got to, which is the solvent front. Because that's key to all your other calculations.
It didn't go much higher.
We don't take, I
Oh,
don't know if it's like wet.
It was probably just.
Now, while we're waiting, I can show you, I think.
Yeah. Interesting how that works. Oh, I'm dumb. The lines. What? Measuring the lines that it creates.
So I'm gonna show you kind of our expected results, just to give you an idea of where we kind of expect some of these bands to be,
No, it's moving.
but the order of the bands we kind of expect.
It's just really slow. Yellow.
All right, so, remember we said yesterday that chlorophyll A would be kind of a blue-green color? Chlorophyll B, we said olive green. And then I don't know if you'll see pheophytin, that is the primary electron acceptor. Carotene usually gets pretty far. And then xanthophylls are usually toward the bottom. So I'm not saying that's what yours will look like, but that's kind of the order they tend to come out. So on the second page of your lab, I gave you a chart with chlorophyll A, chlorophyll B, xanthophyll and carotene. And I put a box for another pigment, like if pheophytin comes out, great. If it doesn't, I mean, the xanthophylls, there's several different ones, so you might just have to pick one of those.
Like teeth and chicken. I want to see how that is. What? Wanda's not gonna be, Jake White's just not there, so Wanda didn't really.
So it's just like.
Oh, I guess I should shut off my recording, huh?
Yeah,
Might help, right?